So, think about the last time you had a terrible bacterial infection.
Wow, yeah, nobody likes that.
Right. Maybe it was, strep throat or a really nasty skin infection, or you were just handed a Z-Pack at urgent care.
Yep, the classic urgent care visit.
Exactly. And you take that little pill, and there is this, like, implicit expectation that you are swallowing a microscopic smart bomb.
That is a great way to put it, yeah.
You fully expect that chemical to just flawlessly hunt down the invading army, destroy it, and completely ignore your own human cells.
Which is actually a huge ask for a pill.
Right. Because when you stop to actually think about it, it's just a mindless, lifeless molecule.
Just a chemical compound.
Yeah. How on earth does a simple chemical know the difference between a harmful pathogen and, well, you?
It is a really profound question. And the answer basically comes down to a concept called selective toxicity.
Selective toxicity.
Okay. Yeah, that is the entire foundation of antimicrobial medicine. It's the art of exploiting the deep, fundamental biological differences between us and them, so that the drug only ruins the pathogen's day.
Well, welcome to today's deep dive, July 25, 2026. Today, we are on a mission to explore the microbiology behind that selective toxicity.
We're getting right into the microscopic details.
We are. We're going to look at the specific structures inside bacteria that these drugs target, the fascinating and honestly kind of heartbreaking history of how they were discovered, and the unresolved debates about how they actually deal the final lethal blow.
And to really understand how we started looking for these biological differences, we have to rewind to a German physician named Paul Ehrlich.
Okay, let's go back.
So around 1900, he coined a term that really set the stage for modern medicine, which was the magic bullet, or Zauberkugel in German.
The magic bullet, I like that.
Yeah, his vision was to find a chemical compound that would behave like a targeted missile, flying straight to the pathogen and leaving the host entirely unharmed.
Which sounds exactly like the smart bomb expectation we have today.
Exactly. And in 1909, he and his assistant, Sahachiro Hada, actually proved the concept worked. They were screening just hundreds of compounds and hit upon compound 606.
Compound 606, very sci-fi sounding.
Right. It became known as salvarsan. It was an arsenic-based treatment for syphilis.
Wait, arsenic?
Yeah, arsenic. Now, it didn't target a specific cellular mechanism the way modern drugs do. It basically just relied on the fact that the bacteria were slightly more sensitive to arsenic poisoning than the human patient was.
So it was just a race to see who died first, the bug or the patient.
Basically.
Yeah.
But it proved the core logic. You can find a chemical that hits the bug and spares the patient.
Okay, let's unpack this, because if you look at our sources for this deep dive, the first true antibacterial drugs that followed Ehrlich didn't come from some clean theoretical understanding of molecular biology.
Oh, not at all.
They came with these incredibly dramatic, almost unbelievable origin stories. It really highlights this massive recurring theme in the history of medicine.
The chasm between knowing a drug works and actually having any idea how it works.
Exactly. Gerhard Domek is the perfect example of that gap.
Oh, absolutely. So in 1932, Domek was working with industrial dyes, of all things.
Like for clothing.
Yeah, industrial dyes. And he discovered that a specific red dye called Promptosil could cure severe streptococcal infections in mice.
Which is wild on its own.
It is, but the leap to using it in humans happened under incredibly desperate circumstances. In 1935, his own daughter, Hildegard, was playing with a sewing needle, pricked herself, and developed a life-threatening strep infection.
Oh, no.
Yeah, and conventional treatments were completely failing. So out of pure desperation, Domek gave her this unproven industrial dye.
And she lived, which is just incredible.
It really is. It launched the era of sulfa drugs.
And Domek even won the Nobel Prize in 1939 for it, though. The political regime in Germany at the time forced him to decline it initially.
Which is a wild piece of history on its own.
Seriously. But even more famous than Domek is, Alexander Fleming in 1928.
Oh, the classic moldy Petri dish story.
Yeah, returning from vacation to find that a stray mold, Penicillium notatum, had landed on his Petri dish and just dissolved the staph bacteria growing there.
The quintessential accidental discovery, right?
Mm-hmm.
But people often forget that Fleming's discovery just sat there for over a decade.
Wait, really?
It just sat there.
Yeah. He knew the mold produced something that killed bacteria, but he couldn't isolate the chemical. It wasn't until Howard Florey and Ernst Chain at Oxford University managed to purify penicillin in 1940 that it became a usable drug.
Wow. which leads directly to one of the most tragic stories in the material we reviewed.
The case of Albert Alexander.
Right. The policeman in 1941, he had been scratching around in his garden and got a tiny cut from a rosebush thorn.
A single thorn.
A single thorn. And that tiny scratch led to a bacterial infection so severe he lost an eye and was on the brink of death.
It's terrifying how fast that used to happen.
Truly. So they gave him the newly purified penicillin the very first time it was ever used this way, and his recovery was just miraculous. His fever broke, he sat up, he ate.
But the purification process was so slow, they simply ran out of the drug.
They had such a limited supply, they were even trying to extract unmetabolized penicillin back out of his urine to recycle it.
That is so desperate.
It was, but it just wasn't enough. The moment the drug was gone, the few surviving bacteria, the ones that had endured the initial assault, began to multiply again.
And he relapsed and died a month later.
Yeah. It remains this stark, devastating early lesson in what happens when you apply a partial treatment.
Because you aren't just failing to cure the patient, right? You are leaving behind the most resilient survivors to regrow the infection.
Exactly. It's selective pressure in action.
It's a heartbreaking story. But zooming out a bit, I can't help but point out how wild it is that scientists like Fleming, Florey, and Domack were initiating these medical miracles without knowing what the drugs were actually doing to the bacterial cells.
They were completely flying blind.
To me, it's like successfully flying a 747 across the ocean without having any concept of how a jet engine actually generates thrust.
That is a perfect analogy. You know the plane stays in the air, but the mechanics are a total black box.
Right.
And that biochemical work, to actually look inside that engine, didn't really start yielding answers until the 1950s with researchers like Jack Strawminer.
Oh, Strawminer, okay.
He was one of the pioneers who finally figured out how penicillin was achieving that selective toxicity, which brings us to the first major vulnerability that antibiotics exploit.
The cell wall. The bacteria's outer armor.
Right.
So let's break down this armor.
Sure.
Why do bacteria even need a wall when, human cells do perfectly fine without one?
Well, it basically comes down to internal pressure. Bacteria are often swimming in environments where the concentration of water is much higher outside their single-celled bodies than inside.
Okay, so osmosis.
Exactly, osmosis. Water constantly wants to rush into the bacteria. They have enormous internal osmotic pressure. Imagine a water balloon that is constantly being hooked up to a faucet.
It's gonna pop.
It's gonna pop. If they didn't have a rigid, tightly woven mesh wall surrounding them, made of a substance called peptidog lichen, they would swell up and literally pop.
But human cells don't have that problem.
Nope. Human cells don't face that same environmental pressure in our bodies, so we never evolved cell walls.
Which makes it the absolute perfect target for selective toxicity.
100% perfect.
I mean, if you have a drug that destroys cell walls, it can't possibly hurt a human cell because there's no wall there to destroy.
And the mechanism is quite elegant. Let's look at beta-lactam antibiotics, which includes penicillin and cephalosporins.
Okay.
These drugs have a specific chemical structure called a beta-lactam ring. Now, when a bacterium is building or repairing its wall, it uses specific building blocks, notably a molecule called D-ala-D-ala.
D-ala-D-ala. Got it.
Yeah. The bacteria rely on special enzymes, often called penicillin-binding proteins, or PBPs, to cross-link those building blocks together and make the wall strong.
And here's where the drug pulls off this microscopic bait and switch, right? Because the beta-lactam ring of the penicillin structurally mimics that D-ala-D-ala building block.
It looks just like it.
So I picture it like handing a bricklayer a fake Lego brick that's been covered in industrial superglue.
That is exactly what it is. The enzyme goes to grab the building block, grabs the drug instead, and its hands are permanently glued shut.
And the bricklayer is completely incapacitated, so the bacteria can't maintain the wall. Water rushes in from that osmotic pressure you mentioned, and the cell just explodes.
Boom. That is exactly what happens.
Wow.
And because we don't have those walls or those specific enzymes, penicillin quest drugs are generally incredibly safe for us.
But the vulnerability has a crucial catch, if I recall from the notes.
It does. That superglued Lego brick mechanism only works if the bacteria are actively building the wall.
Oh.
If a bacterial cell is dormant, if it isn't growing or dividing, the penicillin just flits on by.
Oh, that makes sense. If the bricklayer's on a coffee break, handing them a rigged Lego brick doesn't matter because they aren't trying to build anything anyway. A researcher named Joseph Bigger actually documented this phenomenon way back in 1944. He called these dormant bacteria persister cells.
Persister cells.
Yeah. They effectively go to sleep, sail through a whole course of penicillin completely unharmed, and then wake up once the drug is gone.
So that's why some people get chronic, relapsing infections that seem impossible to clear.
Exactly.
Now, the sources also talk about vancomycin, which attacks the exact same wall-building process, but it uses a completely different strategy, right?
It does. Vancomycin doesn't bother jamming the enzyme. Instead, it physically binds to the Diala building blocks themselves.
Oh, so it attacks the bricks, not the brick layer.
Yes. It basically wraps the actual bricks in duct tape so the bacteria can't use them.
And its origin story sounds like something right out of Indiana Jones.
Doesn't it?
It was discovered in 1953 from a soil sample sent by a missionary working in the deep jungles of Borneo.
And the crude extract they pulled from the dirt was so unrefined they nicknamed it Mississippi mud. Mississippi mud. But it became this vital, last resort drug for highly resistant infections like MRSA.
It absolutely did. So that's the cell wall. But if some bacteria are dormant, or if they evolve to protect their wall, we need to get inside the cell.
We need to attack the factory floor.
Exactly. And this takes us to protein synthesis, and specifically the ribosome.
Okay, the ribosome.
Ribosomes are the microscopic machines inside every cell that read genetic instructions and assemble proteins.
And everything a cell does requires proteins.
Everything. So here, selective toxicity relies on a much more subtle structural difference. Bacteria use ribosomes called 70S ribosomes, which have a slightly different shape and size compared to human ribosomes, which are larger 80S ribosomes.
70S for them, 80S for us.
Right.
So the goal is to design a wrench that only fits into the gears of the 70S machine while slipping right through the 80S machine.
Streptomycin is a classic example of this wrench. It binds to a specific part of the bacterial ribosome and essentially causes it to misread the genetic code.
So like a factory worker trying to read a ticker tape of instructions, but someone smudged all the letters.
Exactly. The bacterium starts producing garbled, completely non-functional proteins, and it dies.
And streptomycin was a monumental breakthrough.
Oh, huge.
It was discovered in 1943 and became the very first effective treatment for tuberculosis.
But the human drama behind it is intense.
Very intense. The discovery came from a soil bacterium found by a microbiologist named Selman Waxman and his graduate student Albert Schatz.
Waxman is actually the guy who coined the word antibiotic, by the way.
Oh, really? I didn't know that. But Schatz was the one doing all the dangerous hands-on lab work with the live tuberculosis bacteria.
Working with live TB in the 40s was no joke.
Not at all. Yet when the 1952 Nobel Prize rolled around, Waxman was the only one on the podium.
And the fallout was incredibly bitter. Schatz ended up suing Waxman.
Wow, a grad student suing his PI.
Yeah, he eventually won formal co-discoverer credit and a share of the royalties in a settlement. It's still taught today as a landmark case in scientific credit disputes.
It just shows human ego is universal, even when you're curing TB.
Definitely.
But going back to those ribosome factories, I have to push back on something, the research.
Okay, go for it.
The sources mention drugs like macrolides and chloramphenicol that also jam up the bacterial ribosomes.
Right.
But if our human ribosomes are 80S, and the drugs only target the bacterial 70S, why do these drugs sometimes cause severe side effects in humans? The notes mention things like hearing loss or severe bone marrow issues like aplastic anemia.
That is a great question.
Right. If the selective toxicity is based on a physical difference, why are we getting hurt?
What's fascinating here is this incredible evolutionary twist. Our main cellular ribosomes, the ones floating around in our cellular fluid, are indeed ADS.
Okay.
But human cells also contain mitochondria, the powerhouses that generate our energy, right?
The mitochondria.
And our mitochondria retain bacteria like 70S ribosomes.
Wait, because billions of years ago, mitochondria used to be free-living bacteria that got swallowed by our ancestral cells.
That is the leading biological theory, yes.
Go. That is mind-blowing.
They are an ancient relic of our evolutionary origin. So the selective toxicity isn't perfectly clean. When you take these antibiotics, they can sometimes cross into your own cells, recognize the ancient bacterial signatures on your mitochondria, and attack them.
So our own evolutionary history literally dictates the side effects we experience today.
It does. It's evolutionary baggage.
That is absolutely crazy. Okay, so we've breached the cell wall, we've sabotaged the protein factories. What about the blueprints themselves?
You mean targeting DNA and RNA?
Exactly.
This is where drugs like the fluoroquinolones come in, which includes ciprofloxacin.
Right.
When a bacterial cell divides, it has to unpack and copy its DNA. Think of taking an old coiled telephone cord and trying to pull it apart.
Oh, it gets super coiled. It twists tighter and tighter into knots.
Right, so bacteria use specific enzymes called DNA gyrase and topoisomerase the 5 to act like microscopic scissors and tape. They snip the DNA to relieve the tension and then tape it back together.
And the fluoroquinolones block the tape, but not the scissors.
Effectively, yes. They bind to those enzymes, meaning the DNA gets cut to relieve the tension, but it can't be put back together.
Oh, wow.
The bacterial chromosome just shatters into pieces, which is immediately lethal. There's also rifampin, another major TB drug, which blocks the enzyme that transcribes DNA into RNA, meaning the cell can't read its blueprints at all.
It's just systemic sabotage.
Yeah.
Now, I also want to ask about targeting the cell membrane.
Yeah.
Not the rigid cell wall we talked about earlier, but the flexible lipid envelope that actually holds the cell's guts inside.
Right, the actual membrane. There are drugs that bypass internal processes entirely and just destroy that envelope.
Like daptomycin.
Yes, daptomycin inserts itself into the membranes of certain bacteria and literally punches wide open pores into it.
Just punches holes in it.
Yeah. The cell's essential ions just leak out into the environment and it dies almost instantly.
Now, the sources also bring up colistin when talking about membrane targets. And this really stood out to me.
I'm not surprised.
The text says colistin was discovered decades ago, but it was largely shelved by doctors because it causes severe kidney toxicity in humans.
It's very toxic, yeah.
So if it's that toxic to us, why on earth are hospitals pulling it back off the shelf today?
If we connect this to the bigger picture of modern medicine, they are doing it purely out of desperation.
Desperation.
Colistin is being revised as a drug of absolute last resort against gram-negative superbugs.
Oh, man.
We're talking about nightmare bacteria like carbapenem-resistant Enterobacteriaceae. The tragic reality is that bacterial resistance has completely closed off our better, safer options.
The bugs have mutated.
Exactly. So doctors are forced to go back into the historical archives and use older, imperfect, toxic weapons, risking kidney damage just to keep the patient alive.
That really sets the stakes for where we are right now. Okay, let's hit the final major vulnerability, metabolism, specifically the folate pathway.
Folate, or vitamin B9, is crucial for making DNA.
And humans need it, but we get it entirely from the food we eat, right?
Right. We absorb it through our diet. Bacteria, however, cannot absorb folate from their environment. They are forced to manufacture it internally, from scratch, through a specific chemical assembly line.
Which is a huge structural vulnerability.
Massive.
If you shut down the assembly line, they run out of building blocks for DNA.
And this is where those sulfa drugs Domak discovered come back in. They mimic a molecule called PABE to block step 1 of that folate factory.
And then there's another drug, trimethoprim, that blocks step 2.
Right. And the sources mention that doctors will often prescribe them together as a drug called cotrimoxazole to create what's known as a sequential blockade.
A sequential blockade?
It is an incredibly effective strategy. By hitting the bacteria twice in the exact same metabolic pathway, you make it exponentially harder for them to evolve resistance.
Because if they mutate to survive the first block, the second one still catches them. So let's take a breath here. We have these 5 major targets, right? Bursting the cell wall, jamming the ribosome factory, shattering the DNA blueprints, punching holes in the membrane, and starving their folate metabolism.
Those are the big 5.
And we used to think of these drugs in as strict binary, like they were either bactericidal, meaning they killed the bacteria outright, or they were bacteriostatic, meaning they just hit the pause button, freeze the bacteria in place, and wait for your own immune system to show up and clean house.
Right. That was the traditional teaching.
But our notes suggest that strict binary is actually pretty outdated.
Very much so. Modern microbiology sees it as highly conditional. Whether an antibiotic kills or just pauses depends on the dose, the specific bacterial species, and even the growth phase of the bug.
Really?
Yeah, the drug lanazolid, for instance, acts as a bacteriostatic freezer against staph infections, but it can act as a direct bactericidal killer if you use it against strep infections.
Wow. So the context completely changes the weapon.
It does.
And speaking of how these weapons actually kill, this part blew my mind. We've been mass producing and swallowing these drugs for nearly a century, but the scientific community still doesn't fully agree on the actual mechanism of death.
It is a brilliant reminder that textbook science is never truly settled. It's always evolving.
Tell me about the 2007 hypothesis.
So in 2007, a researcher named James Collins published a highly influential hypothesis. He looked at drugs that hit totally different targets, beta-lactams hitting the wall, fluoroquinolones hitting the DNA, and suggested they all ultimately share the exact same lethal downstream pathway.
The idea was that no matter what you break, the resulting stress causes the bacteria to produce reactive oxygen species.
Basically, toxic oxygen radicals that cause the cell to poison itself from the inside out.
A unifying theory of antibiotic death.
Exactly. But then in 2013, James Imlay's lab published a counter-study challenging the whole concept.
What did they find?
They argued the methodology used to measure those oxygen radicals was flawed. They maintained that the sheer structural damage, the broken wall, the shattered DNA, is enough to explain the cell's death without needing a secondary oxygen poisoning mechanism.
It's still a live scientific argument. The textbook isn't closed.
Not at all.
And speaking of things people get wrong, I mean, if you're listening to this and wondering why your doctor won't give you a Z-Pack for your flu, let's clear up some major common misconceptions.
First and foremost, antibiotics absolutely do not work on viruses.
Right. And if you recall the 5 targets we just discussed, the biological logic is undeniable. Viruses are fundamentally different entities.
They don't have cell walls. They don't have 70S bacterial ribosomes. They don't synthesize folate.
There's literally no physical structure for the antibiotic to target.
None.
Yeah.
So taking an antibacterial drug for a viral head cold doesn't just do nothing. It actively exposes your own healthy gut microbiome to selective pressure, encouraging resistance for no reason.
Which just accelerates the superbug problem.
Yeah.
Another misconception that shocked me was about prescription lengths. We've all had it drilled into our heads, always finish your entire course of antibiotics, even if you feel better.
Yep. The golden rule.
We treat it like a sacred rule to prevent superbugs. But a 2017 analysis in the BMJ really challenged that orthodoxy.
They did a deep dive into the evidence behind the always finish the course rule and found it was surprisingly thin.
Really? Then how?
In fact, they argued that for some specific infections, taking antibiotics past the point where your symptoms resolve might actually increase the selective pressure that drives bacteria to mutate.
It is wild.
Now, public health guidance is slowly becoming more nuanced, though obviously anyone listening should follow their doctor's specific advice based on their unique illness.
Of course. But here's where it gets really interesting, though.
Okay.
When we talk about antibiotic resistance, we have this massive guilt complex.
We do.
We think of it as a modern human invention. We messed up by overprescribing, we dumped it in livestock feeds, we created these superbugs. But the sources point out that researchers found antibiotic resistance genes in DNA pulled from 30,000-year-old Beringian permafrost.
Oh, and the timeline goes back even further than that.
Wait, further than 30,000 years.
A 2012 study isolated bacteria from La Chiguilla Cave in New Mexico. This is a cave system that had been geologically sealed off from the surface world for roughly 4 million years.
4 million years.
And the bacteria they found inside were already resistant to multiple modern antibiotics. They even had defenses against synthetic drugs that didn't exist in nature.
How is that logically possible if humans weren't around to prescribe them?
Because antibiotics are fundamentally ancient chemical weapons.
Soil microbes fighting each other.
Exactly. Soil microbes live in incredibly crowded, competitive environments. They have been using these chemicals against each other in a microscopic arms race for eons to fight over resources.
So one fungus produces a chemical to kill its neighbor, and the neighbor evolves a microscopic pump to spit the chemical back out.
Or an enzyme to chop the chemical in half. Human medicine didn't invent this warfare. We just crashed a party that's been raging in the dirt for millions of years.
We just figured out how to bottle their weapons.
Pretty much.
Which brings us to why all of this matters right now to the person listening. Because that ancient arms race is catching up to our bottled weapons in a terrifying way.
The stakes could not be higher for global health. A major 2022 analysis published in The Lancet estimated that bacterial antimicrobial resistance directly caused over 1.2 million deaths worldwide in 2019 alone.
1.2 million.
Yeah. And the O'Neill Review, which was a major report commissioned by the UK government, projected that if current trends continue unchecked, resistant infections could surpass cancer as a leading global cause of death by mid-century.
It's a numbers game. Every time an antibiotic is misused, whether someone demands it for a viral cold or it's dumped into agricultural feed just to make livestock grow a little faster, it applies selective pressure.
It wipes out the weak and leaves only the strongest, most resilient bacteria.
And those survivors are terrifyingly efficient. They can literally trade resistance genes with each other, like trading cards, a process called horizontal gene transfer.
They can hide in protective slime layers called biofilms.
And our weapons just stop working. The 5 targets we outlined today, the wall, the ribosome, the DNA, the membrane, and the folate, that is a finite list. It's a rapidly shrinking list of vulnerabilities.
And every single time a new, highly resistant strain emerges, it closes off one more exploit on that list. It forces us closer to a post-antibiotic era where a simple scratch from a rose bush could once again become a death sentence.
Thank you so much for joining us on this deep dive. We've covered a lot of ground today, from desperate fathers trying unproven dyes to the precise microscopic machinery of cellular destruction.
A massive topic.
But I want to leave you with one final thought to mull over as you go about your day. We started by marveling at how a mindless chemical pill knows how to seek and destroy an invader in your body. But if bacteria have been waging this chemical warfare in the soil for millions of years, inventing both the weapons and the resistance genes long before humans even walked the earth, what unimaginably powerful antimicrobial targets and terrifying resistance mechanisms are still lying completely undiscovered in the dirt right beneath our feet?